Hey everyone, in this video we're going to be conducting the experiment for Lab 9.1, which is phenotypic variation. And phenotypic variation is basically a way some cells will regulate their gene expression based off of environmental factors. We're going to use Serratia marcescens, which has the ability to produce a red pigment at one temperature, and it does not produce the pigment at the other temperature. So I'm simply going to do a streak plate of Serratia and incubate it at 37 degrees Celsius, and we will incubate the other one at room temperature. So phenotypic variation can be observed in Serratia marcescens. It's one of the few bacteria that can produce a bright pigment, most commonly a red pigment. Serratia marcescens is commonly found in the intestine and is often found in the soil, and it causes about 2% of nosocomial infections of the bloodstream, lower respiratory tract, urinary tract, surgical wounds, skin, and soft tissues of adult patients. You may have observed Serratia marcescens at home if you don't wash or clean your shower curtain very often. That pink color is most likely due to Serratia marcescens. The pigment produced by Serratia, it offers protection against excessive UV light from the sun, serves as an antibiotic, and it enhances flagella production. It also has cytotoxic qualities. You can read more about Serratia in the textbook within Chapter 9 to help you answer the questions in the lab report. So right now there's two streak plates. One's going to be incubated at 37, one at room temperature, and we will see which temperature the red pigment is produced. So here's the results from Lab 9.1, Serratia marcescens Phenotypic Variation Lab. Here on the left we have the 37 degree plate showing a mostly whitish cream color colony, a little bit of pink to it. And then on the right here we have the room temperature plate where the bacteria produce the reddish pink pigment. So based off the environmental conditions, the bacteria here on the right with the colder temperature activated the gene for that pigment production. And here on the left, the bacteria keeps that gene off based off of the warmer temperature. So what you'll do is you record the observation of the growth for number 1, week 2. Now for number 2, week 3, what we can do is we can pick a colony off of the room temperature plate, put it on a new plate, and incubate it at 37 degrees, and take a colony off of the 37 degree plate and put it at room temperature, and we can see what those results look like. So I'll go ahead and set that up right now. So what I'll do is I'll pick a colony off the 37 degree plate, put it on a new plate, and we'll incubate this one at room temperature. So I'll do the same streak plate technique to get isolated colonies. And I'll take one of these pink colonies and put it on a plate, and we'll incubate that one at 37 degrees. Alright, so this is the week 3. This is for answering question number 2. On the left now we have the room temperature incubation from the 37 degree plate. So it's a switch. So here we see we have the pigment produced. And on the right we have the 37 degree from the room temperature plate. I spelled plate wrong. And here we see that that pigment is not produced. A little bit of pink to it, just like before. So again, the gene is activated at room temperature, and it's kept turned off at 37 degrees Celsius. So record the observations on number 2 in the lab report. And this will conclude the Lab 9.1 lab.